Okay. And main pointer. First of all, I will introduce Marchantia because this is also a model plant for lower plants. Fiscomitrium patens is a moss. It's a model plant for mosses, and Marchantia polymorpha is a model plant for liverworts. There are additionally hornworts as the third group, but there is actually no model organism within hornworts. The studies are too preliminary to select a model plant for them. So what are key features of Marchantia? These are descendants of the earliest land plants. So Devonian time in Paleozoic, this is the time when first liverworts could be observed. So really old. And then it's in its life cycle, haploid form is the dominant one. So we had to develop a haploid genetics for it. Then it's vegetative propagation by Jimmy and self-propagates isogenic lines. So I will show you, they produce some small Jimmies that they can grow to adult tally. And on part of that, if you will segment tally, they can also regenerate. So this is easy to regenerate. We also have isogenic lines. It means if they are haploid, they are isogenic. So easy to propagate in vitro, and they are small, so easy to cultivate. Gametes induced are by far-red light, so they have some photoreceptors that are sensitive to far-red light, and if you give far-red light, you will get production of sexual organs. And crossing of them is easy. They easily regenerate in tissue culture as well. They have high frequency of nuclear and plastic transformation if you transform them. And the genome is really small, like 280 megabase. Sequence has been completed and annotated. And this is the web page for genome sequence, genome annotation, transcriptomic data, and so on. Marchantia info, www. And here you can see thallus of Marchantia. These are Gima cups. Inside you have small Jimmies. When they are mature, they fall out and germinate into new thalli. Here you see a female sexual organs. On the bottom, sporophytes will develop. And here you can see a wood of Marchantia thalli producing female, I think, gametanchophores, or male. Are these male? I think male gametanchophores. So now, this is a complex thalloid liverwort. So this is the Gima cup, and these are Jimmies, small Jimmies. Here you see male sexual organs, and here the sperm cells should move. Wait a moment. Is it? I am not able to move them. Oh, they move. They move. Okay, so this is rather exceptional for plants, that liverwort produce mobile sperm cells with two flagella that have to reach egg cell in separate, grown on separate thalli, because the liverworts are dioecious, what means that they produce male and female vegetative thalli. So here you see a female gametangiophores, and here you see a single archegonium and with egg cell inside. Then these are sporophytes, so they are not yellow, they are not green, they are yellow and completely dependent on haploid green tissue. And these are spores. So within sporophytes, meiotic, after fertilization, meiosis takes place, generating spores, and spores can germinate into new haploid thalli. So this is the life cycle. So sperm cells, dispersal, and zygote formation at the bottom of a digit of archegonium, And here are single archegonia with sperm cells. You have diploid, you have meiotic divisions, and sperms germinate into male and female vegetative thallis, producing female or gametangiophores, archegoniophores containing eggs, and sperms that are motile. And here you can see, you can put on top of the anteridiophore, so producing male sperm cells, a drop of water, and then sperm cells are released. You can collect them in a tube, and then you can put the drop of water containing sperm cells on the bottom or even at the upside of the archegonial disc, and then you will get sporophytes. And here you can see small spores germinating here. They are in a magnification. And here are GEMA cups with GIMIs for vegetative propagation. So before I will start, I should also say that the genome is small, and the number of genes is strongly reduced. For instance, SPL transcription factors are unique for plants, but in Arabidopsis you will have, I may guess, like 40 SPLs, maybe even more, divided into 4 clades. But in Marsantia polymorpha, you have just 4 SPL transcription factors. Each is the founder of the clade. So, and in many cases, you have this strong reduction of gene number. So this is also nice to have the possibility to follow the fate of duplications, changing functions, and so on during land plant evolution. So I will start with microRNA that was isolated from Pellia endivifolia, and this is a completely different group because Marsantia belongs to Marsantiopsida, while Pellia belongs to Jungermanniopsida, and their common ancestor was living like 250 million years ago. And we started with this species because it grows very easily in Poland. Marsantia is also everywhere in Poland. And this is also dioecious, and these are male thallus producing antheridia. They have no these stalks. They are just sticked to the thalli surface. And we were the first to identify microtranscriptome of liverworts. This was published in 2015. And we identified there are more than 40 novel, unique for liverworts microRNA families. And among these, we identified one that we called 8185. And then it turned out that it was also published to be present in Marsantia polymorpha, and the name was 11889. And since then, I will call it 118899, since this is Marsantia number. Complicated, but what to do? So if you see at the expression of this microRNA, you can see that it is almost not expressed in male vegetative thallus. It is expressed in male vegetative thallus containing antheridia, and it is not expressed almost in female vegetative thallus, and it is not expressed in female thalli producing archegoniophores. So in Pellia, these are NGS data. It is almost exclusively present in, let's say, vegetative thalli with antheridiophores. And then we found that this microRNA is quite similar to the Marsantia one, but surprising for us, it is also similar to Chlamydomonas reinhardti. This is algae. So till this time, there were no common microRNAs between land plants and algae, and we were the first to show that there are some common microRNAs between algae and land plants. So then we shift to Marsantia polymorpha because genetic tools are nicely developed for Marsantia polymorpha, and found that this evolutionary conserved microRNA is exclusively expressed in sperm cells. So you can see here, these are male vegetative thalli. These are only antheridiophores. This is female vegetative thalli. These are archegoniophores. These are NGS data. It is only present in sexual organs of male male has sexual organs. Northern also looks beautiful. This is the only band for anteridiophores here. But anteridiophores had different parts, so we were interested to know whether it is present in sperm cells or maybe in the surrounding of sperm cells or maybe in stalk. So we isolated anteridia and we purified these single parts and found that in sperm cells, this is the amount of microRNA, but this was not done by using IgL and NGS, but Tachman probe. Do you know what are Tachman probes? But not everybody, I understand. So Tachman probe is a probe where you have a stem and loop structure to which you design a primer that is complementary to your microRNA, and then using RT-qPCR, you can measure the amount of your microRNA. So these are sperm cells. These are archegonia. These are not archegonia, sorry. These are anteridia. So anteridia containing sperm cells. These are anteridiophore remnants, so sperm cells were released. This is male vegetative thalli, and these are anteridiophore stalks. So this was clear that this microRNA is expressed in sperm cells. So then what we did, we using CRISPR-Cas, all tools are there. We generated null mutants, KO mutants, for microRNA 11889. These are these transgenic lines with deleted microRNA sequence, so it cannot act. And we looked for some phenotypic features. We were a little bit disappointed, but you have to wait. So this is wild type thalli, and this is the mutant with popping out anteridia. This is the only one we found, and also that anteridia are bigger than in wild type. So this is calculation is a little bit bigger than in the case of wild type plants. However, in these mutant lines, we also tested the amount of microRNA, and it is not present. It is present in wild type plants, but it is not present in both KO mutant lines. So we looked for the target of this microRNA, and using degradome data, you know already everybody about it, we found one target. This is DASP12 mRNA. DASP12 means dual specific phosphatase. So it dephosphorylates tyrosine and serine threonine as well. So this is dual specificity phosphatase. Somehow weird target for microRNA, but yeah, the result is a result. Yeah. So since the hybridization is not very strong, and somehow I'm getting a feeling that hybridization of liverwort, or at least Marchantia microRNAs to their targets is not as strong as in higher plants. But to get, really good, convincing results, we need more examples. This is just my feeling based on several other examples as well. So this is the hybridization between microRNA and its target. So to be sure that this is indeed the target, because T-plot was very nice with encoding sequence, we also performed 5' raise to look for 5' ends within degradome, let's say, and we identified for 13 isolated and sequenced colonies. This is the product that we cloned and sequenced. 8 were exactly finishing at the nucleotide we were aware is the place of cleavage. Yeah? And then we also found that if you look at the level of DASP12 in male vegetative thalli and in anteridia, it is dropped down in sperm cells where microRNA is present. This is wild type. So we know that microRNA is in sperm cells, and the level of DASP12 is dropping down. And in the mutant, the reverse. This is the wild type in sperm cells, while this is the mutant with DASP12. So microRNA cannot target this mRNA, and it is highly upregulated. On top of that, We also constructed plants overexpressing DASP12. And phenotype was very similar. So with this anteridia popping out of the surface of anteridial disc. And we also did Western to see the protein. And surprisingly, we used antibodies, human antibodies, because it turned out that DASP12 is present in all eukaryotic until studied cells. So it's in human, in higher plants, in fungi, in Saccharomyces cerevisiae, it's everywhere. So, and the structure, as I will show you later, is also very conserved. So the antibody, when we looked where for which part of, no, I think these were how do you call it when you have many epitopes, polyclonal antibodies, not monoclonal, but polyclonal antibodies. So there was a big chance to get good results. And indeed, if you look, this is a male anteridiophores where there is no DASP12 in wild type. This is wild type archegoniophores, a little bit. Then you have DASP12 KO mutant and 2 lines overexpressing DASP12. So you can see that DASP12 is in high amount. Yeah? Okay. Then we wanted to see whether knockout or overexpression of target, knockout of microRNA overexpression of target will affect sperm cells somehow, structure, movement, and so on. And till now, we still do not know much about structure, a little bit. We are waiting for some microelectron images. But these are, you can see here, the movement of wild-type sperm cells. So they go somehow directly, yeah, as a straight line. If you look at the KO mutant of microRNA, they are somehow lost. So they do not know exactly how to go. And if you look at DASP12, the phenotype is even stronger. So they efficient than wild-type sperm cells. Okay, then we looked into the structure using fluorescent microscopy. This is propidium iodide, which stains DNA. And you can see this is the shape of the sperm cell in wild-type plants. I should add that sperm cells in Marchantia are almost all filled with nucleus. And then they have one mitochondria on the top, let's say here, one mitochondria where flagella are, and one chloroplast. So practically the whole body is filled with nucleus. But then when we look at the mutant, KO mutant, and overexpressing lines, we found that the structure of this of the DNA with the nuclei is different. It is somehow fragmented. And can you see it? That it is not so smooth like in wild-type plants. Yeah, it is somehow fragmented. More strongly in overexpressing plants. I should add that to overexpress target, we use target with mutation, that it cannot be cleaved by microRNA on these lines. So this is insensitive to microRNA target. Yeah? You know, when we overexpressed in the background of wild-type plants, DASP12, we introduced a copy of mRNA with changed sequence, so amino acids remained the same. But the sequence was changed that microRNA cannot cleave it anymore. Yeah? So what we did next, we remembered a paper published in 2017 on overexpression of protamines. in Marchantia polymorpha. Protamines are proteins that are involved in packaging of DNA in sperm cells. This is for animals typical, this is for liverworts typical. In higher plants like Arabidopsis, you have no protamines, you have specific histones that do that, but not protamines. So I don't know whether you can see it here, but it is also fragmented. When you have, it's not well? Oh, this stopped working. Yeah. Okay, so maybe I will try to be more close to the micro. So you can, I don't know whether you can see it, but this is fragmented as well. Maybe I can enlarge it. Can you see it now? That it is fragmented? Oh. I don't know what I did. I can see it here. Okay. Or now maybe like this. So now it is better visible, yeah? Yes. That the DNA with the nucleus is also fragmented. And this is overexpression of proteins. So we immediately tested the amount of protein in mRNA, and then we also did proteomic studies of KO mutant for microRNA over expression of DASC12 and wild type to look for proteomic comparison. And what we found, if you look at mRNA level, this is wild type, this is KO mutant of microRNA, this is overexpression of DASC12 mRNA. It is up, however, we were a little bit surprised that this is not as high as in KO mutant. Nevertheless, if you look at proteomic level, you can see this is wild type level of protamine. This is only one gene for protamine in Marchantia. This is KO mutant, this is overexpressing. So somehow DASC12 controls the level of proteins, yeah, that are involved in DNA packaging in sperm cells. So what we know about packaging of DNA, this is the spermatogenesis from first spermatids and or spermatocyte through different stages until you reach the mature sperm cell. And if you look at the histones that are involved in packaging of DNA at different stages till mature sperm cells, and these are so-called toroids. These are not solenoids packaging, these are so-called toroids. They pack much more tightly DNA, at least in human, let's say. Yeah? Nobody knows how it is exactly in Marchantia, but in humans, these are toroids. But here are human histones responsible for packaging of DNA during spermatogenesis, and I marked in yellow their orthologs found in Marchantia that are upregulated in KO and overexpressing mutants. So something with packaging is going on. So these are also results of this quantitative proteomic analysis that revealed altered protein profiles in antheridia and sperm cells. Antheridia were sperm cells still maturate. Ah, but I forgot that I should not move. Sorry. It was extensively used because it was exchanged at the first day. And patents give up. Thank you very much. Is it on? Yeah, I think so. So here you see again the antheridia that are not mature and sperm cells of microRNA mutant, KO overexpressing of white. So generally I will not go into the details because we are still working on. So this is not published, I'm just showing you recent results. But what was typical for that in antheridia, the majority of these proteins was downregulated, while some histones that were downregulated in antheridia are upregulated in sperm cells, in mutants. Okay, so now something more about DASP12. It has a nuclear cytoplasmic localization. You can see this is nucleus, this is cytoplasm, and this is in the Nomarsky field that you can see DNA in the nucleus and cytoplasm. And here is a comparison of sequence between different organisms. You are not able to see it. But here you have, with the use of AlphaFold, the structure of DASP12 in different organisms. So this is Marchantia. I have to come close because I don't see it. This is Marchantia. This is human. So look, the tertiary structure is almost completely identical. Then this is yeast here, almost identical. Now, surprise, surprise, this is Arabidopsis thaliana. And if you would see here at the comparison of these sequences, you would find that in Arabidopsis thaliana, phosphatase domain is missing. It's deleted. If you take another species like Arabidopsis lyrata, it's there. But in thaliana, it disappeared. But this part is still conserved between Marchantia, human, yeast, and Arabidopsis thaliana as well. This is when we, jak mówimy nakładamy, when we, no merge, or how do we call it in English? Superimpose, thank you. Superimpose structures from all these organisms. You can see it is really very similar. So then we got interested in this part that was kept in Arabidopsis, because in the meantime we got knockout mutants of this and overexpressing mutants in Arabidopsis, and we have really severe phenotypes. So something is in this C domain, and this C domain turned out to contain 2 zinc fingers. And this is shown here, 1 zinc finger, second one, here it is, and I think here it is. And if you make some, if you look at the charge of the surface in this domain, you see some positively charged patches that may somehow suggest that it can bind to DNA. And as you remember, probably I always told you that microRNAs mainly target transcription factors. Okay, so then we looked at the expression profile of DASP12 in different organisms. So this is human DASP12, and these are normal cells, but then you see when spermatogenesis is taking place, so it is required, DASP12 is required, but constantly drops down, and at the end, in late spermatids, it's not present. DASP12 is not present. When you look at Arabidopsis, we do not have such a development of microspores, but you can see that if you have flower, rosette, and so on, in stamen, it is downregulated. Yeah? In anthers, this should be anthers. I don't know, shoot, flower, rosette leaf, and this is for sure antheridia. Maybe I lost it. Anthers. Maybe I lost it. And then this is Marchantia, and again you see gamelings, so young plants, Gima caps, some parts of mature thalli. You see antheridiophores here, and sperm cells. They do not contain DASP12. So the profile that you need DASP12 probably at the beginning of spermatogenesis, but then you lose it somehow, is typical for human. It looks like for Arabidopsis and also for Marchantia. So these are mutants, pollen grains of Arabidopsis, DAS12 overexpressing, and DAS12 mutants, and they are strongly deformed. So this is wild type of Arabidopsis. This is overexpression. The pollen is really affected, and this is delta DASP12. This is also affected. You can see it easily that they are probably dead, because we cannot get homozygous plants. So it is deleterious for sexual propagation. So then we got a little bit of mRNA from Human Institute for Human Genetics. They gave us RNA from biopsy of men suffering from obstructive and non-obstructive azoospermia. I do not know exactly what does it mean, obstructive, non-obstructive azoospermia, but they have problems with producing active sperm cells. And they have reduced levels of DAS12 in testicular tissues. This is wild-type men, and these are men that suffer from of this azoospermia.
Azoospermia, I think that they cannot move.
Oh, they don't cannot. But what is obstructive and non-obstructive? I don't know. I don't know this. Never got into the details. Nevertheless, if they suffer for this azoospermia, what means that they do not move, right? Yeah. So you have less DASP12. So something is on. We of course we have to now to get into closer cooperation in the case of human, but this is not very easy because we are a plant department. We have of course people working on human cells as well, but we have also in the Institute of Human Genetics people working on spermatogenesis, so this would be the easiest way to try to cooperate with them, because we desperately looked for microRNA in Arabidopsis that would potentially target DASP12, and we were unsuccessful. So we looked for novel microRNAs mainly, so there are deposited data for small RNAs from microspores, from the vegetative nuclei, generative nuclei, and there are a lot of small RNAs, and I tried to identify microRNAs, but none of them seems to target DASP12. So it looks like it is very typical for liverworts, at least for Marchantia. But in the case of human, we identified 2 or 3 microRNAs that potentially could target DASP12 in human. But remember, this is the seed sequence that counts, yeah, in the case of the hybridization between mRNA and microRNA. So we have some potential microRNAs that are upregulated in human sperm cells and may target DAS12. But for that, we need cooperation with this department. And it is not done. So coming back to Marchantia, we wanted to know how mutants of delta microRNA and overexpressing of DAS12 will exhibit efficiency in fertilization, and we found strong reduced in fertilization efficiency. So this is wild type when you cross it. These are spores, and you can see sporophytes. And then when we, let's say, fertilized 100 wild type Archegonia, we got 60 sporophytes. But then when we crossed, now I have to come closer, delta microRNA as female and delta microRNA as male, this is here, only 9 sporophytes were found. This is percent, sorry, 60% was here found as producing active sporophytes, here only 9%. When we took female delta microRNA and male overexpressing DASP12 line, we had like 14% of sporophytes formed. Then when we took female overexpressing line and delta microRNA, we got 8%. And when we took DASP12 overexpressing and DASP12 overexpressing in both, we got like 3% of fertilization efficiency. And if you look now at these sporophytes, they do not look like the wild type. So they eventually somewhere you find sporophytes that produce spores, but they very often are within tetrads. So they somehow do not separate in single spores. Sometimes you get some sporophytes that are at the beginning green, but they do not develop, and even they may die. So, and this is the worst example where we very rarely found a single sporophyte that was able to produce spores. But if they produced, they germinated. Yeah? So it is not that it completely abolishes the production, the sexual fertilization, but the sexual fertilization is strongly reduced. So conclusions are that studies on model organisms bring unexpected universal discoveries. That microRNA module is required for proper anti-radiophos development and efficient sexual reproduction. And DAS12 is an evolutionary conserved protein across eukaryotes and affects male gamete development. And with that, I would like to finish, but just I want to conclude that I mentioned that maybe DAS12 is a transcription factor. And why do I think it is like that? First of all, that microRNAs very often target transcription factors. It has cytoplasmic nuclear localization. It is in really small amounts. Our pain is that when we did proteomic studies, we cannot find DAS12. So you saw the Western for overexpression, but to do it, we had to precipitate several times proteins from isolated from tissues, and to have them more to do Western. Transcription factors generally are present in low amount in the cells. And on top of that, what I didn't show you, we have also finally got mutants with downregulated expression of DASP12. We were not able to get Marchantia plants using CRISPR-Cas with knockout of DASP12. So we finally generated artificial microRNAs that will target DASP12, and we have two independent lines with strongly downregulated DASP12 level, but still vivid. And in these lines, proteins are downregulated, while in overexpressing lines, it is upregulated. So you see some direct relation between the level DASP12 and potential target, who knows, DASP12. Protamines in that case. So what we think now we have to do, and we are preparing ourselves to do chromatin immunoprecipitation. I will talk about it also in my next talk. So we have constructed lines with DAS12 fused to GFP. Then if it binds to chromatin, we will cross-link chromatin using UV or formaldehyde and we will get proteins covalently bound to DNA. Then we will fragment chromatin, and then we will immunoprecipitate using anti-GFP antibodies. And first of all, I think we will do qPCR to find enrichment maybe of promoters of protamines. This is my first goal. And the second one is another transcription factor. Maybe this is a sandwich like transcription factor, protamines. Or if we will be unsuccessful and my idea will fail, we will do DNA libraries from a chip and look for eventually bound DNA fragments to understand what is going on. So this is the work done with that genome editing is a transformation tool for studying plant metabolism and development, and CRISPR-Cas9 has revolutionized research in lower plants, and studying lower plants provides evolutionary insights and novel metabolic pathways. And with that, I would like to thank you. This is our department, Faculty of Biology, and Halina Pietrykowska is the main executor of all studies. Wojtek Karłowski is bioinformatician, and Artur is co-PI of all these studies. With that, I thank you very much for this time. We will have another lecture again. And I am open for questions, of course.